my name is Maggie Shostak, and I am a first-year PhD student with East Carolina University in North Carolina, and I am part of the eDNA projects. I work with Dr. Aaron Field with eDNA, which stands for Environmental DNA, and what we do is we take samples from different environments, so whether it's the water, a shipwreck, and from those samples, when we process it, it'll tell us what microbes are living there, what fish species have passed through the area, and we can determine whether or not the ecosystem is healthy by how much biodiversity we have. So here in Saipan, we're focusing mainly on the World War II wrecks, so that includes shipwrecks, plane wrecks, and tanks. And so what we do is we have a dive team that goes underwater, and we take a conical tube, which is just a plastic tube to hold samples, and a scraper, and we scrape off biofilm, which is the fluffy stuff you see on shipwrecks underwater. It can look orange, it can look white, it has a whole range of colors. We take that, scrape it into the conical tube, and then at the surface, we process them. So we take the excess water out because we only care about the biofilm at the very bottom. From there, we're gonna send the samples back to our lab at ECU, and that's where I will extract DNA from the samples using a kit, specifically a Power Soil Pro kit from Huygens. And after I extract the DNA, I send it to a sequencing center in Nova Scotia, Canada. It's the Integrated Microbiome Resource Center, IMR. And they send me back all the DNA sequences, and I can then use different programming sources such as RStudio and a Data2 pipeline to determine what the taxonomic classification is of the samples that I have. So eDNA has become a relatively new emerging technology for research. One great analogy we use is you can go out fishing and you might not catch the specific endangered species that you're looking for, but if you take a scoop of water, its DNA is going to be in that water, so you'll know for sure that that species is present. So it really helps in terms of time. It's a lot easier to take a scoop of water and look at its DNA to determine what species are present compared to actually going and specifically counting everything. For the shipwrecks, they are corroding due to being underwater, especially in salt water, which the salinity is very high, and that has been shown in other studies to cause corrosion to accelerate. And these sites are very important for not only local veterans and CNMI economy and blue economy. So by understanding what microbes are present, we can determine and extrapolate an idea of how fast these shipwrecks, plane wrecks, and tanks are corroding to see if there's something we can do to help keep them preserved, whether that's limiting the amount of tourism on the sites in terms of like touching. So every time you touch something, you're leaving your DNA on that object, and that can actually influence how well it's preserved throughout time. I would recommend for kids to try everything they can. Get out there, anything to get you introduced to all the different areas of science that would then help you determine maybe you want to join a biology group, or maybe you want to become an archaeologist. So try your best to be open to all these new ideas, be open to trying new things. If you think you might not like it, the more you have experience with different stuff, the more likely you are to actually find that you really enjoy it.